Hi guys, happy Friday. Today I am at the fitness fair, so I'm outside chaperoning the fitness fair. So Mr. DeSutter is going to be with you today. So today you're actually going to get to skip number 1. We're going to do that the next time we meet. And you're going to go straight to number 2, which is the photosynthesis review. And then number 3, which is complete part 1 of the plant raw materials. And that's a group lab. Sorry, my computer's being a little funky right now. So let's go ahead and go to the classroom and open up the photosynthesis review. Here, you're going to basically click through the slides and answer the questions. So it's just review, use your models, take your best guess, and then click turn in. You are welcome to do the slide and then chat with each other about what you got. That might be helpful. The last 2 are the hardest slides, and we'll go over them together after you have completed. Actually, we'll go over them together next time I see you. So we'll do that I guess I'll see you on Tuesday. Okay, so Mr. DeSutter, if you want to pause this video and have the students complete their photosynthesis review. Okay, now that you've done your photosynthesis review and you took your best guesses there, next you're going to go to the other assignment listed today, which is called Plants Raw Materials. And you are going to be doing part 1. And to do this, you're going to basically look at 4 different variables and see how they affect the rate of photosynthesis. So today you'll be investigating how does the amount of light affect photosynthesis? How does the amount of carbon dioxide affect photosynthesis? How does the color of light, that's an interesting one, affect photosynthesis? And then how does the temperature affect photosynthesis? So how can we measure photosynthesis? Well, here's kind of a cool way that it's measured. Let me play this little lab video. Play, there we go. Okay, so what you can see here is, get this out of here. You can see that,
as shown earlier, pondweed can also be placed in a beaker under an upturned.
You have a aquatic plant that goes through photosynthesis, and you have a funnel that has a cap on it turned upside down, and then a light source. Watch what happens. Can you see what's going on? Yeah, there's bubbles. What's in those bubbles? Air. What kind of air? What kind of air would there be if a plant was placed in light? Yeah, oxygen. So you could either count the number of bubbles, or you could measure the space. The more bubbles, would the space increase or decrease? The more bubbles, the space would increase. So that's a way that you can measure photosynthesis, because oxygen is one of the outputs of photosynthesis. So let's go ahead and start with a little bit of predicting. So if there's an increase in the amount of CO2, which is over here, what do you think it will do to the rate of photosynthesis? Do you think it will increase or decrease? Delete the incorrect word, or the one you're not predicting. Then says the greater the rate of photosynthesis, the more or the less oxygen is produced. So if the rate of photosynthesis is higher, will you have more or less oxygen? Okay, and then the final one is, if there's a low number of oxygen bubbles, what can you say about photosynthesis? Low number of oxygen bubbles. What can you say about photosynthesis? Okay, so now that you got some predictions down, we're going to go to part 1. So what you need to do is have one person in your group click where it says click here and share it with the members in your group. So you're going to be working with the people at your tables. Mr. DeSutter, if someone's absent and someone is working alone, just have them join another group. So go ahead and pause the video and give them a moment to share out. Okay, so now you are on the lab, and this is what you're completing today. I don't know why it's doing this. Okay, So what you're going to do here is there's 4 different variables. You can see they are denoted by the topic slide. So this one, the first variable you're going to look at is the color or the wavelength of light. So what you're do first, the computer's being funky, is you're going to, on that first slide right here, pre-experiment claim, what color of light, red, orange, yellow, green, blue, will produce the most oxygen? So erase all the ones you think won't, just a prediction. Okay. So we'll kind of do this first one a little bit together. So this will be the easiest one. So there's 4 variables. If you're in a group of 3, the person that gets 2, they'll do this first one because it's the easiest probably. If you're a group of 2, just divide them 2 and 2. Okay, so then we're going to use the gizmos or the Explore Learning that we have used. We used this when we did How Does Light Enter the Eye? When we switched around and you could put your tongue on a ball or you can and see how the sensation goes through your body. So you're going to click there on slide 2, click on where it says gizmos, and then you're going to need to come to this website and log in. When you created your login, so click login, you used your username that was in front of @wacohigh.net. So mine is LL England. You should have put @wacohigh.net. So once you're here, you'll click on that link on slide 2 where it says gizmos. You will need to log in, and the login that you created in my class was whatever you have in front of @wacohigh.net. So mine is LL England, and then your password. Log in. Now, if you have any trouble getting logged in, you do have 2 options. The first thing that you could do is you have a free trial for 5 minutes. So there's a possibility you could actually get this done in 5 minutes because I can show you pretty quickly how to do it. Or you could actually ask a classmate. You can give your computer to a classmate and have them log in for you. Just don't look at their password. So that's what I would have you do in this case because I'm not going to be there to reset your password. If neither of those 2 things are an option, you can email me. I'm at school. It's just whether or not I'll see the email in time. But this should work for you because everyone who had me, I told you to use your school login and password. So then you're going to click Launch Your Gizmo. And like I said, I'm going to demonstrate got it fixed. For the color or the wavelength of light, you're going to click on this tab here. This is color. And then you need to do 10 data points. So you're going to keep everything constant. So I'm going to keep the temperature constant, the light intensity, the CO2 levels constant. Just don't mess with them. Don't even touch them. The only thing I'm going to change is down here, the color of light, which relates to the wavelength of light. So I need 10 data points. The first thing I need to see is the range. Okay, the smallest wavelength that's available is 400. The largest is 700. Okay, so 700 minus 400 is 300. I need 10 data points. How do I figure out what to do? Well, 300 divided by 10 is 30. So what does that mean? To get 10 data points, starting at 400, click on where it says table. I'm going to start at 400, click record data, and then I'm going to go up to 430, click record data, and then I'm going to increase by another 30, 460, record data, keep going up by 30, 490, record data. You're going to continue to do this until you have 10 data points. You're trying to use this data to determine which wavelength of light does photosynthesis, the highest amount of photosynthesis occur. So once you have 10 data points, you can see I only have 1, 2, 3, 4. And you can see that everything else is kept constant. The intensity of light is constant. The temperature is constant. And the CO2 is constant. I'm going to transfer these numbers to my data table on my slide. And then on the next slide, it asks me to insert my graph. Well, that's really easy to do. You just click over here where it says graph. And then I want to make sure that my graphs is graphing the wavelength. So you have to click on what you want on the x-axis, which should be your independent variable. So in this case, I'm adjusting the wavelength. So I click on wavelength. Then I come over here to the right-hand side where it says camera. And it's actually going to take my graph, and I can just click copy image. And then I can go to my assignment and paste it where it says insert graph here. So Control V, and I can paste my graph. And then all you have to do to finish your variable is answer the questions. What trends can be seen? What's the best level? And then describe what you did. Was your pre-experiment claim supported? And the last question is, what is your takeaways? Okay, so you're going to complete this for all of your variables. Then, once your group is done, so if you're a group of 3, one person's going to do 2. So I recommend having the first person do the light, and then the second or the same person do carbon dioxide. So if someone volunteers to do 2, have them do the first 2. If you have 2 people in your group, no problem. 2 people each do 2. And then once you have that done, you're going to go back to your original assignment where it says share out your takeaways, which is part 2. Just share out your takeaways. So it looks like this. What did you learn about each of these ideas? Okay, so that's your stopping point. Part 3 we'll do in class when we get back. So go ahead and start on your individual gizmo part. So, okay, thank you. Have a good weekend, and I'll see you on Tuesday.